The development and maintenance of an organism is orchestrated by a set of chemical reactions that switch part of genomes off and on at strategic times and locations. Epigenetics is the study of these reactions. A great example of epigenetic phenomenon would be your skin, eyes, teeth, hair, and organs. All of these have exactly the same DNA genetically speaking. You cannot tell my skin from my eyes, yet they look very different. This difference has been termed epigenetics. It isn't directly related to genetic changes by the way we utilize these different genes, so the process can be so different that it causes a profound difference that one tissue looks like skin and the other looks like an eye.
There are two kinds of modifications that can affect DNA. One is DNA methylation, which involves the addition of a methyl group to the 5 positions of the prime pyrimidine ring. The other is a key event where DNA is wrapped around a series of proteins called histones. The DNA that holds on the histone very tight and is hidden from view from the cell. Imagine the ability to turn off cells such as cancer cells.
It isn't easy, but it's effective. It's important not only to turn the right genes on, but also turn the right genes off. Histones and chromatin components have key roles in the decision-making process. But although there are many different problems, the benefits are endless. Not only can this be used for cancerous cells, but it can be used for diseases like HIV. The side effects for epigenetic therapy are minimal, nothing like the side effects of things such as chemotherapy. Epigenetic therapy is the future of the fight against cancer and still has much more headway to make. It has the possibility of leading the future of medicine into an age of no cancer, among other diseases, plus not to mention future job opportunities. If you count age as how many times the stem cells divided, the cancers are awfully old tissue. If you think of a 60-year-old patient, the epigenetic changes in the cancer would reflect the actual age of the DNA, which might be 200 or 300 years, depending on how long the cancer has been dividing incessantly in that particular case.
The idea of epigenetic therapy is to stay away from killing the cell. Rather, what we are trying to do is diplomacy, to change the instructions of the cancer cells. Cancer cells start out as normal cells. They have the set of instructions that is present in every one of our cells. The reversible nature of epigenetic aberrations has led to the emergence of the promising field of epigenetic therapy, which is already making progress with the recent FDA approval of 3 epigenetic drugs for cancer treatment. In this review, we discussed the current understanding of alterations in the epigenetic landscape that occur in cancer compared with normal cells. The roles of these changes in cancer initiation and progression, including the cancer stem cell model, and the potential use of this knowledge in designing more effective treatment strategies.